neurologic, hepatic, and renal syndromes. In the United States, foodborne diseases have been estimated to cause 6 million to 81 million illnesses and up to 9,000 deaths each year. Quite often in the literature, we meet the terms food and waterborne infections, or food-related diseases. Three categories of food poisoning can be mentioned. Foodborne disease can be mentioned. Infections or parasitic infestations acquired by eating food contaminated with microorganisms or parasites. Next group, clinical problems that result from eating food contaminated with toxins. With toxins of bacteria. And then third group, clinical sequel from eating inherently poisonous animals, plants, or mushrooms. Major infectious causes of acute diarrhea include different types of agents. For example, bacterial agents, first bacterial group, bacterial agents such as Salmonella, Shigella, Yersinia, Campylobacter. These all are bacterial group. Next, viral group, such as adenovirus, astrovirus, leucovirus, Norwalk virus or noroviruses, and rotavirus. So group of viral diarrheal diseases. Next, parasitic or protozoan diarrheal diseases. Such as Balantidium coli, Cryptosporidia, Entamoeba histolytica, Giardia lamblia, and others. And also food poisoning group, which include such sources of bacterial toxins as Bacillus cereus, Clostridium perfringens, Staphylococcus aureus, Vibrio parahemolyticus, and in some conditions, Salmonella species also can cause disease like food poisoning. Here you can see bacterial causes of foodborne diseases with their food sources. And you can see that some of these bacteria produce toxins in food. Such as Bacillus cereus, Staphylococcus aureus, and In the others, ingestion of bacteria is important. In case of Salmonella, Shigella, and Campylobacter. General information of pathogenesis of food poisoning. All microbes causing food poisoning have general property. They are capable to produce exotoxins which also we call enterotoxins of various types. Despite of differences in the mechanisms of their action, so they have some differences depend of bacteria which release this exotoxin. But despite of differences, all of them stimulate hypersecretion of water and electrolytes from the intestinal epithelium and thus produce watery diarrhea. As a rule, these toxins as I said, these toxins begin to be developed by microbes in food. Before food ingestion. And it is explained extremely short incubation period at food poisoning. In some cases, less than 1 hour. Characteristic of enterotoxin. Enterotoxin has exotoxin protein nature, and it is released from viable bacteria. Enterotoxin has localized action. Which is confined to particular cell types or cell receptors. Generally, all exotoxins have localized specific action. And target of enterotoxin is enterocytes. Yes. The prototypical enterotoxin is cholera toxin. Composed of 1A and 5B subunits. While the B subunit binds holotoxin, to the enterocyte surface receptor, the ganglioside GM1, then the fragment of the A subunit is translocated across the eukaryotic cell membrane into the cytoplasm. Where it catalyzes and causes persistent activation of adenylate cyclase. The end result is an increase of cyclic AMP in the intestinal mucosa. Which increases chloride secretion and decreases natrium absorption. leading to loss of fluid and to production of isotonic diarrhea. There are head labile and head stable exotoxins. They have intermediate resistance to pepsin digestion. And it's important that some enterotoxins have super antigenicity properties. Based on the unusual non-antigen specific activation of T cells. Pathogenesis of food poisoning include ingestion of the food contaminated with microbes and especially their toxins. Which then faces the normal gastric acid barrier and then some of them get intestine, small intestine, and then enterotoxins activate secretion of fluid and salts into the lumen of the small intestine and lead to secretory diarrhea. Limited duration of action of enterotoxins up to 48 hours. Causes the short-term course of food poisoning. So enterotoxins have limited concentration, limited duration of their action. Yes, and then that's why food poisoning has short-term course. As the microbes causing food poisoning have no invasive properties, the role of microbes is not important for food poisoning. So inflammatory reaction in the intestine is expressed weakly. At stable enterotoxins with super antigenic properties. Sometimes insert into systemic blood flow and cause pyrogenic effect. So sometimes patients with food poisoning can demonstrate intoxication. Online students, your conference will be interrupted in some 7 minutes. Please connect using the same link, okay? And we will continue.
Okay.
Okay. Now about clinical characteristic of food poisoning. Short incubation period is common for food poisoning because patients get already toxins in food. Poisoning begins abruptly with nausea, vomiting, crampy abdominal pain, and diarrhea. As I said, the diarrhea is usually non-inflammatory. So there are no blood, no mucus. It's true. And diarrhea is of lower volume than that in cholera, or in case of toxigenic Escherichia coli infection. Generally, fever and rash are absent in food poisoning, and the patient is neurologically normal. The majority of cases of food poisoning are self-limited and resolve between 8 and 24 hours after onset. But in severe cases, hypovolemia and hypotension can develop. Here, characteristics of selected foodborne illnesses are demonstrated. And you can see that 4 diseases caused by Staphylococcus aureus, Bacillus cereus, Clostridium perfringens. Fever is uncommon, but possible, of course. But it's not specific, it's not common symptom. Now we will discuss some of food poisoning in more details. First one is staphylococcal food poisoning. Okay, we continue. Staphylococcal food poisoning. Although most cases probably do not come into medical attention or are not diagnosed, staphylococcal intoxications, staphylococcal food poisoning is the second or third leading cause of diagnosed food poisoning in Western countries. Staphylococcal food poisoning is a gastrointestinal illness caused by eating foods contaminated with toxins produced by the bacterium Staphylococcus aureus. About 25% of people and animals have Staphylococcus on their skin and in their nose. So they are carriers of Staphylococcus aureus. It usually does not cause illness in healthy people. But in some conditions, Staphylococcus has the ability to produce toxins, to produce exotoxins, enterotoxin, that can cause food poisoning. Nearly all strains of Staphylococcus aureus secrete enzymes and cytotoxins. which includes hemolysins, nucleases, proteases, lipases, hyaluronidase, and collagenase. So different enzymes to convert local host tissues into nutrients required for bacterial growth. Some strains of Staphylococcus aureus and it is important for, especially for food poisoning. Some strains produce additional toxin, exoprotein, enterotoxin, which is important for us for our today's topic, and other toxins also. Such as toxic shock syndrome toxin, the exfoliative toxin A and B, and glycoside. Each of these toxins is known to have potent effects on cells of the immune system. They also have other biological effects as well. Now, epidemiology of staphylococcal food poisoning. How a patient can get this food poisoning. It is caused by eating food containing enterotoxin of Staphylococcus aureus. Processed meats and custard-filled baked goods are common food vectors for staphylococcal food poisoning, perhaps because staphylococci can tolerate conditions of high protein, high conditions of salt or sugar, and so grow without competition in these environments. Food is contaminated by Staphylococcus aureus when it is prepared non-hygienically, by human individuals who are carriers of these organisms. Carriers of Staphylococcus aureus. Cutaneous or nasal carrier. Contaminated preparation equipment and poor personal hygiene of food handlers are frequently implicated as well. And so if food is contaminated by these carriers with Staphylococcus, then bacteria can multiply in the food and produce enterotoxin. And staphylococci can multiply at a wide range of temperatures. Thus, if food is left to cool slowly and remains at room temperature after cooking and remains for a long period of time, the organisms, Staphylococcus, will have the opportunity to form enterotoxins. So high temperature and food, they are special condition for Staphylococcus aureus, which lead to releasing enterotoxin. Foods that are not cooked after handling, such as sliced meats, puddings, pastries, or some sandwiches, are especially risky, but if only contaminated with Staphylococcus aureus. So never leave perishable food out for more than 2 hours, or even 1 hour, if it's hotter than 90 degrees Fahrenheit outside. And it's important, you have to know that food contaminated with staphylococcal toxin may not smell bad or look spoiled. The disease has a high attack rate and is somewhat more common during the summer than at other times of the year. And it's common not only for this disease, but for all food poisoning or foodborne diseases, as you know. The staphylococcal enterotoxin is head stable thus tolerating cooking conditions that kill the organisms that produce them. So Staphylococcus aureus is killed by cooking, but the toxins are not destroyed and will still be able to cause illness. Clinical characteristics of staphylococcal food poisoning. It is characterized by a sudden start of nausea, vomiting, and stomach cramps. Most people also have diarrhea. Symptoms usually develop within 30 minutes to 4 hours after eating or drinking an item containing staphylococcal toxin. And the symptoms last no longer than 1 day. Severe illness is rare. and can lead to severe dehydration. And occasionally, but staphylococcal food poisoning sometimes is fatal. Especially in the very young, the very old person. And people weakened by long-term chronic illnesses. Staphylococcal food poisoning cannot be passed from one person to another. So carrier of Staphylococcus aureus can be source of infection for other person, but person with staphylococcal food poisoning cannot be source of infection. See? You can suspect staphylococcal food poisoning based on the type of symptoms and their fast resolution. Although laboratory tests can detect toxin producing staphylococcus in stool, or vomit, and in foods, but these tests are usually not ordered except during an outbreak. And treatment of this patient, the main part of treatment is rehydration of patients. Now, food poisoning syndromes caused by Bacillus cereus. Bacillus cereus is a common soil saprophyte. It is a gram-positive spore-forming, motile, aerobic root that also grows well anaerobically. Bacillus cereus causes two different types of food poisoning. The emetic type emetic. And the diarrheal type. For both types of food poisoning, the food involved has usually been treated and surviving spores are the source of the food poisoning. Bacillus cereus is not a competitive microorganism, but it grows well after cooking and cooling. The head treatment will cause spore germination and in the absence of competing flora, Bacillus cereus grows well. Generally, growth of Bacillus cereus in food can usually be inhibited by refrigeration. Although some strains of the organism can grow slowly at 4 degrees Celsius. And in some cases, frying before serving may not destroy the preformed head-stable toxin. Online students, we will be interrupted once more in 10 minutes. So we will take a short break, maybe 15 minutes, yes? And then you can connect us using the same link, okay?
Okay.
Okay.
Well, so Bacillus cereus food poisoning is underreported, as both types of illness are relatively mild. So two types of illness, yes, two distinct food poisoning syndrome are caused by Bacillus cereus. Emetic, when nausea and vomiting occur approximately 6 hours after eating toxin-contaminated food. Yes, and diarrheal type. When diarrhea occurs approximately 20 hours after eating toxic-containing food. So one type with vomiting and nausea, and other type with diarrhea. And both types of illness are relatively mild and usually last for less than 24 hours. So it is common for all food poisoning, short-term course. But sometimes, however, occasional reports have described a more severe form of the diarrheal type of Bacillus cereus food poisoning. Now about Clostridium infection. Clostridium are gram-positive, spore-forming, obligate anaerobes. They are ubiquitous in nature. Universally present in soil. Some of them are pathogenic for humans. Particularly under anaerobic conditions. In humans, Clostridia reside in the gastrointestinal tract and in the female genital tract. Although they occasionally are isolated from the skin or the mouth. Vegetative self of Clostridium perfringens possesses at least 20 possible virulence factors. Including active tissue toxins and enterotoxins. Clostridia may cause different types of infections, including superaerative deep tissue infections, skin and soft tissue infections, bacteremia, and also intestinal syndromes such as food poisoning, Enteritis necroticans, and antibiotic-associated colitis. Food poisoning caused two species, Clostridium perfringens and Clostridium botulinum. Clostridium perfringens food poisoning is the second or the third most common cause of food poisoning in Western countries. Human carriers should be considered as a possible source of contamination for Clostridium perfringens type A food poisoning. The food sources primarily involved are meat and poultry. Strains of Clostridium perfringens that contaminate meat manage to survive initial cooking. During reheating, the organisms sporulate and germinate. After the contaminated food has been ingested, Clostridium perfringens organisms sporulate in the small intestinal lumen. The bacterial cells then lyse, releasing spores and enterotoxin, which is responsible for symptoms of this type of food poisoning. Clostridium perfringens can also survive high temperatures. About Clostridium food poisoning. What else? As we said, if the food is served without reheating to kill bacteria, live bacteria may be eaten. And the bacteria also produce a toxin inside the intestine that causes illness. Now, clinical characteristic of Clostridium food poisoning. incubation period 8 to 24 hours. After ingestion of foods heavily contaminated with the organism, with the Clostridium perfringens. After ingestion, toxin is produced in the intestinal tract. Symptoms are epigastric pain, nausea, and watery diarrhea. Usually lasting 12 to 24 hours. Fever and vomiting are uncommon. Diarrhea appears to be caused by a het labile protein enterotoxin. Which lead to secretory diarrhea. The illness is self-limited, rarely lasting for more than 24 hours. And is rarely fatal. More severe form of diarrhea Clostridium perfringens causes among elderly, debilitated individuals, and it is associated with antibiotic use in hospitalized population. In this form of disease associated with antibiotics, diarrhea is generally more profuse, of longer duration, and accompanied by abdominal pain. Blood and mucus have been detected in the feces of the affected patients. we will discuss salmonella infection, general characteristic, pathogenesis, clinical characteristic, and management, treatment, and protocols. So, Salmonella is a genus of the family of Enterobacteriaceae. It is a major cause of diarrheal diseases. In both developed and developing countries. The genus Salmonella is named after Daniel Salmon, an American veterinarian. Daniel Salmon first isolated Salmonella cholerae suis from pigs with hog cholera in 1884. Salmonella are effective commensals and pathogens that cause a spectrum of diseases in both humans and animals. And animals, including domesticated and wild mammals, reptiles, birds, and even insects. General characteristic of Salmonella, they are gram-negative, non-sporulating, facultatively anaerobic straight rod bacteria. Salmonella.
Please, doctor, can you get someone who fix it the window because it's so cold here? And it's so cold here coming in this place.
Yes. From window, yes?
Yes, you
Yes,
can.
yes. Yes, I believe I see, because the same problem we have in our room also. I don't know, we can. You can just change your place now, but then I just say this problem. I will say about this problem.
Okay, just
For us, and we try to solve somehow.
I will check the second one.
Yes, the same. I see this problem from the same. No, he is not. He is not.
Last week is interesting.
This is not very great because in our room it's more, it's worse. We try to think something, I don't know how, but maybe just mechanical, yes. Okay, now, Salmonella are motile. They can move using peritracheal flagellum. Only approximately 1% of organisms are able to ferment lactose and therefore may not be detected if only McConkey agar or other semi-selective media are used to identify Salmonella. Based on assay for fermentation of lactose. The differential metabolism of sugars can be used to distinguish many Salmonella serotypes. For example, Salmonella typhi is the only organism, only Salmonella that does not produce gas on sugar fermentation. Salmonella nomenclature has undergone many changes in recent years. They are classified into serovars serotypes based on the lipopolysaccharide O, flagellar protein, and sometimes the capsular antigen. There are more than 2,500 known serovars. Within a serovar, there may be strains that differ in virulence. According to the latest naming system, there are 2 recognized species in the genus Salmonella: Salmonella enterica and Salmonella bungorii. A proposed 3rd species, Salmonella subterranean, was identified in 2005 but has not been universally accepted. Salmonella enterica has 6 subspecies that are identified by name or by number. You can see in table, Enterica 1, Salemi 2, Arizona 3A, and so on. The complete formal names of these serovars are all shortened. For example, Salmonella enterica, subspecies enterica, serovar enteritidis, can be called just Salmonella enteritidis. Salmonella bongori, as well as subspecies 2, 3A, 3B, 4, and 6 are rarely isolated from clinical specimens. Therefore, almost all Salmonella organisms that cause disease in humans and domestic animals belong to Salmonella enterica subspecies enterica 1. As I said, Salmonella strains can be classified on the basis of the O lipopolysaccharide surface antigen and H flagellar antigen into serogroups, serotypes, and serovars. Some strains have somatic VI antigen and capsular T antigen, mucose M antigen. And according to World Health Organization data, Salmonella typhimurium and Salmonella enteritidis are most commonly isolated in clinical practice.
This one shows our last statement. Serotyping of all surface antigens can be used for formal identification. Most laboratories perform a few simple agglutination reactions that define specific O antigen into serogroups, designated as groups A, B, C1, C2, D, and E Salmonella. Strains in these 6 serogroups cause approximately 99% of Salmonella infections in humans and warm-blooded animals. Although this grouping is useful in epidemiologic studies, it cannot identify whether the organism is likely to cause enteric fever, or diarrheal disease, because considerable cross-reactivity occurring among serogroups. For example, Salmonella enteritidis, which typically causes gastroenteritis, and Salmonella typhi, which causes enteric fever, they are both group D. And similarly, another frequent cause of gastroenteritis, Salmonella typhimurium, and some Salmonella paratyphi, another cause of enteric fever. They are both group B. Today we will discuss just about non-typhoidal salmonella infection. Yes, non-typhoidal salmonella are leading cause of foodborne diseases in humans worldwide. While gastroenteritis is the most common presentation in healthy adults, but systemic disease can also occur, particularly in those who are immunocompromised, including children and the elderly. Focal intraintestinal, extraintestinal infections can also be seen. Salmonellosis is generally self-limited in healthy people. Although long-term side effects can occur. In many countries, the incidence of human salmonella infections has increased markedly. In the United States, the incidence rate of non-typhoidal salmonella infection has doubled in the last 2 decades, with an estimated 1.4 million cases occurring annually. The incidence of non-typhoidal salmonella infection is highest during the rainy seasons in tropical climates and during the warmer months in temperate climates. And coinciding with the peak of foodborne outbreaks. Some Salmonella serotypes, such as Salmonella typhi and paratyphi, cause enteric fever. They are highly adapted to humans and have no other known natural hosts. So Salmonella typhi and paratyphi only human bacteria. They are only human bacteria. Others such as Salmonella typhimurium, enteritidis, typically causes gastroenteritis. They have a broad host range and can infect a wide variety of animal hosts and humans. Some Salmonella serotypes, such as Dublin and Arizona, Dublin in cattle, Arizona in reptiles, they are mostly adapted to an animal species and only occasionally infect humans. Salmonella species are mainly transmitted by the fecal-oral route. And people are often infected when they eat contaminated foods of animal origin. Sources of salmonella infection. The most common sources of salmonella infection are eggs and poultry. Online students, again, you will be interrupted in 10 minutes and you connect using the same link without break. The most common sources are eggs and poultry, sometimes cattle, pigs, rarely pets and wild animals, cat, dog, rodents, wild boars, deer, cold-blooded animals, reptiles, and sometimes human, when sick or carer. Salmonellosis associated with exotic pets, including turtles, iguanas, lizards, and snakes also. Exposure to pet birds, pet rodents, dogs and cats, and to pet food and pet treats made from animal parts are other potential sources of human salmonellosis. In chicken, infection localizes into the ovaries and upper oviduct tissues and is transmitted to the forming egg before shell deposition, resulting in contamination of the albumen and yolk. Cooking eggs until all liquid yolk is solidified kills Salmonella enteritidis. So the use of pasteurized egg products remains the safest alternative for institutions and the general public. Both outbreaks and sporadic Salmonella enteritidis infection have been mostly associated with egg and poultry. as well as sporadic cases associated with international traveling and exposure to birds and reptiles in home too. Human or animal feces may contaminate water and the surface of fruits and vegetables and may not be removed by washing. So in this case, fruits or vegetables can be also cause of infection. Pathogenesis. Salmonella infections begin with the ingestion of bacteria in contaminated food or water. The median dose required to produce disease with approximately 10 in 6 level bacteria. The acidic pH of the stomach reduces the number of ingested microorganisms then enter the intestinal tract. And sometimes more than 99% of ingested coliform bacteria are killed within 30 minutes but in case of normal gastric acidity. But sometimes Salmonella display an adaptive acid tolerance response that probably facilitates bacterial survival in the stomach and passage to the small intestine. Salmonella can penetrate the gut epithelial layer by 3 routes. First left panel demonstrates that Salmonella adheres to and enters M cells, which it then kills by causing apoptosis. Having penetrated the epithelium, it infects macrophages and gut epithelial cells. In the middle panel, you can see that Salmonella invade gut epithelial cells directly by adherence of their fimbriae to the luminal epithelial surface. And third, right panel, dendritic cells sampling the gut lumen protrude dendrites between epithelial cells. These effectively breach the epithelial layer and can be infected by Salmonella in the lumen. Here you can see current experimental model of the series of events.
Can you show the previous slide? Thank you.
So now, experimental model of the series of events leading to an inflammatory diarrhea during Salmonella serotype typhimurium infection. So in first 15 minutes, there is invasion of epithelial cells. Next 1 hour, neutrophil extravasation and increase in vascular permeability, which cause edema. Next 3 hours, neutrophil induced tissue injury, epithelial detachment, and beginning of fluid accumulation. And 8 hours, next 8 hours, effusion of large amount of neutrophils and protein-rich fluid into intestinal lumen. Submucosal and pairs patches macrophages. Macrophage invasion may occur through bacteria-mediated macrophagocytosis or through phagocytosis directly by several receptors present on the macrophage. Available data indicate that the ability of Salmonella to survive and replicate within macrophages is essential for dissemination within the host. And as a result, induction of systemic disease. And in persons with positive blood cultures, so in systemic disease, the majority of organisms are contained within the mononuclear fraction. Patients with diseases that impair host defense mechanisms and increased frequency of severe Salmonella infection and Salmonella bacteremia. Here are some pictures of pathogenesis of salmonellosis. penetration, phagocytosis with macrophages. In case of survival, it leads to bacteremia. Now about risk groups for severe salmonella infection. There are some risk groups. And patients with diseases that impair host defense mechanisms, they are an increased frequency of severe salmonella infection. There are patients with sickle cell disorders, patients with some infectional diseases such as malaria, Bartonellosis, schistosomiasis. Patients with lymphoma and leukemia also are more prone to develop salmonella bacteremia. And recently, prolonged and recurrent refractory salmonella bacteremia has been observed in patients with AIDS. Exotoxin and endotoxin play a key role in the pathogenesis of salmonellosis. The term endotoxin was coined in 1893 by Pfeiffer to distinguish the class of toxic substances released after lysis of bacteria. From the toxic substances, exotoxins secreted by live bacteria. All endotoxin molecules are similar in chemical structure and biologic activities. So endotoxin molecular complex can be divided into 3 regions. Or specific chain, which consists of a variety of repeating oligosaccharide residues. And next, core polysaccharide that forms the backbone of the macromolecule. And lipid A. The polysaccharide portions are responsible for antigenic diversity, polysaccharide portion, whereas the lipid A confers toxicity. Dissociation of the complex has revealed that the polysaccharide is important in solubilizing the toxic lipid A component. This slide demonstrates characteristics of bacterial endotoxins and classic exotoxins. And you can see that endotoxin has lipopolysaccharide nature. Exotoxin is protein. Endotoxin is functioning as a part of membrane, but exotoxin is extracellular, diffusible. Endotoxin is head stable. Exotoxin may be head labile or head stable. Both of them have antigenic properties, but exotoxin is high specificity, so it forms toxoid. Exotoxin usually demonstrates enzymatic activity. Endotoxin has pyrogenicity, which is not common for exotoxin. Biologic activity of endotoxin. Endotoxin acts as a B-cell mutagen, stimulating the polyclonal differentiation, the multiplication of B cells, and the secretion of immunoglobulins, especially immunoglobulin G and immunoglobulin M. They are endotoxins are mitogen. They are stimulating the polyclonal differentiation and multiplication of B cells, which then secrete immunoglobulins, especially immunoglobulin G and M. Lipopolysaccharides or endotoxins activate macrophages to enhance phagocytosis and cytotoxicity. Macrophages are stimulated to produce and release some inflammatory factors, main inflammatory factors, lysosomal enzymes, interleukins, human necrosis factors, as well as other cytokines and mediators. And these are powerful mediators of inflammation and septic shock that accompanies endotoxin toxemia. And here the list of biologic activity of endotoxin. As a result of endotoxin, there are fever, leukopenia or leukocytosis, complement activation, depression in blood pressure, mitogenicity, induction of prostaglandin synthesis, hyperthermia, disseminated intravascular coagulation, human necrosis, hypotension, or even shock, and in large dose lethality. In case of large doses of endotoxin, lethality. And these events can accumulate in sepsis and lethal shock. Here you can see lipid inflammatory mediators, which is also play an important role in pathogenesis of inflammation of salmonellosis. All these enzymes synthesize eicosanoids, prostaglandins, and leukotrienes, and platelet activating factor, that have variable inflammatory consequences. And cause also membrane phospholipids or endotoxins. Also about hemostasis, initial activation of Hagemann factor, which in turn can activate several humoral systems, resulting in coagulation, in activation of the complement alternative pathway, plasmin activation, all these can lead to bleeding and inflammation and hemorrhages. Also, kinine activation releases bradykinin and other vasoactive peptides, which causes hypertension. So the net effect of lipopolysaccharides or endotoxin is to induce inflammation, intravascular coagulation, hemorrhage, and shock. Now, clinical characteristic, diagnosis and treatment of salmonellosis. Salmonella infection proceeds in different clinical forms: localized gastrointestinal forms and generalized forms such as Salmonella bacteremia, like typhoid fever, and Salmonella bacteremia with metastatic infection or septic form. And also Salmonella infection may occur in form of intestinal carrier state. In gastrointestinal form, after an incubation period, which is usually 12 to 48 hours, the illness starts suddenly with crampy abdominal pain and diarrhea. A chill is common. Fever is present in most patients and may reach 40 degrees or higher. Although occasional patients have nausea and vomit once or twice, vomiting is not persistent. The diarrhea may be watery and of large volume or small volume. The stools may contain mucus and occasionally blood. Polymorphonuclear leukocytes are present in the stool. Depending on severity of disease, diarrhea may be mild or may be severe, with up to 20 to 30 stools a day. The abdomen is tender to palpation. Transient bacteremia may occur and is most likely in infants, the elderly, and in patients with impaired host defense mechanisms. Symptoms usually improve over a period of days, with fever lasting no more than 2 to 3 days and diarrhea no more than 5 to 7 days. However, these symptoms may occasionally persist for up to 14 days. More severe disease is seen with malnutrition, inflammatory bowel disease, and in patients with AIDS. Also, reactive arthritis may follow enterocolitis in up to 7% of cases. And it is especially frequent in those patients with the HLA B27 phenotype. Online students, you will be interrupted once more, I think the last time, and connect again once more, and for I think for 5 or 10 minutes only. Okay, just for 5-10 minutes. The last time.
All right.
Okay.
Well, patients with Salmonella bacteremia. Patients with Salmonella bacteremia usually, with syndrome of Salmonella bacteremia, usually complain of fever and chills for a period of days to weeks. Gastrointestinal symptoms in this case are unusual, but in some patients, the syndrome of Salmonella bacteremia follows classic enterocolitis. Other symptoms are non-specific, such as malaise, anorexia, and weight loss. Metastatic infection of bones, joints, mycotic aneurysm, meningitis, pericardium, lungs, heart valves, and other sites are common. As a result, symptoms may be related to the site of metastatic infection. So, symptoms of Salmonella bacteremia are intoxication, and specific symptoms depend on the site of infection. Stool cultures are usually negative for Salmonella, but blood cultures are positive. So any Salmonella serotype can produce the syndrome of bacteremia, but Salmonella cholera suis is most likely over 50% of infected. So Salmonella cholera suis is more likely to produce the syndrome of bacteremia. The asymptomatic intestinal carrier state may result from apparent infection, which is the most common form of Salmonella infection, or may follow clinical disease, convalescent carrier. The carrier state is usually self-limited to several weeks to months, with the incidence of positive stool cultures rapidly decreasing. By 1 year, far less than 1% still have positive stools. A patient who has had salmonella in the stool for 1 year, it means chronic carrier, and it is likely to become a lifelong carrier. The diagnosis of salmonella infection is made by isolating the organism from the stool in enterocolitis. From blood, urine, and stool in enteric fever, and from the local site in localized infection. Also, polymerase chain reaction is used to detect bacterial DNA. Serologic studies are of little clinical value in salmonella infections other than typhoid fever, but they may be of use in epidemiologic studies. A stain smear of the stool usually demonstrates polymorphonuclear leukocytes in patients with salmonella enterocolitis. However, test results are usually come back after symptoms have resolved. So clinical science and epidemiological studies are more important for diagnosis. The primary approach to treatment of salmonella enterocolitis is fluid and electrolyte replacement, so rehydration. Drugs with antiperistaltic effects, such as loperamide, can relieve cramps, but should be used sparingly because they can prolong the diarrhea and intoxication syndrome. And it's important to know indications for antibiotic treatment. They are severe enterocolitis, Salmonella bacteremia, Salmonella enterocolitis, Salmonella bacteremia, immunocompromised patients, it may be elderly patients, infants, patients with severe chronic diseases. Antibiotic therapy for non-typhoidal salmonella infection in adults should be administered as a preemptive treatment. Consider for neonates, persons more than 50 years of age with possible atherosclerotic vascular disease, and patients with immunosuppression, endovascular graft, or joint prosthesis in case of severe gastroenteritis. Consider on an individualized basis for patients with severe diarrhea and high fever who require hospitalization, this patient also, and in bacteremia. So in these cases, antibiotic therapy can be administered as a preemptive treatment. The main group of antibiotic, it's main medicine is ciprofloxacin, and other ceftriaxone. So in severe case, it may be used intravenous. Also, antibiotic therapy should be administered in endocarditis or arthritis, meningitis, and other localized septic infection in salmonella infection, salmonella bacteremia. And also, duration of antibiotic treatment is important also. In severe case, it may be given for a long time. Till 40, more than 1 month, for example. In case of endocarditis or arthritis, salmonella endocarditis or arthritis. What Okay, doctors, that's all for now. And now you will have a break for 40 minutes, maybe, yes? And then you will have your practice lessons. Also, online students, also, you will have break and then connect with your teachers. Okay?
Okay.
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 Thank you.
Thank you. Have a good day.